Well, I had a question about something that I've gotten from CROI. Now, of course, I used to be able to follow CROI without actually having to attend because they did such a good job of posting the talks that I could almost follow along at a slower pace, maybe, but it was great. And this year I feel cut off. And I feel like I'm back in the old days where only the priests learned Latin and only the priests could read the Bible. And we're waiting for the priests to tell us what to think and all that. But I hear about these HTI vaccines, the vaccines. And can you speak to that a little bit? What might have been talked about at the conference and what they are? These therapeutic vaccines. Do you know what I'm talking about?
Yeah, I saw this is a talk, a late breaker, I think, from Beatrice Moth or Mothe. I'm not sure how you pronounce it. And it was a phase 1/2 placebo-controlled trial, and I had to look up what HTI stood for actually just before this too, because I couldn't remember. So it's basically it's a viral vector immunogen, but the HIV insert is basically it's derived from over 50 epitopes, T cell epitopes targeting both CD4 and CD8 T cells that are what they say restricted by a wide range of HLA types, which means that hopefully it would be beneficial for a lot of different people from different genetic backgrounds. And if those epitopes are relatively conserved, so that if one of them is mutated, it's associated with a big fitness cost for the virus. So most likely, the idea is to have sort of a broad consensus type of sequence, so that the T cells that are generated that are specific for HIV by this vaccine would be most likely to be targeting the infecting virus or the viruses that are in, the reservoir. And so what they did is it's a really complicated vaccine regimen. So they had, it's 3 just straight up DNA immunizations, 2 MVA with this HTI insert, 2 Chad Ox, so the chimp adenovirus vector that also is used in the COVID vaccines, and then followed by 1 more MVA. So it's really very, a large number of immunizations. And then there was a treatment interruption as a part of the trial. And they said it was safe, well tolerated, did induce strong immune responses. And in terms of the kind of main readout, which was time to viral rebound and rebound set point in this 24-week treatment interruption period, the time to rebound didn't really differ, but they did claim a kind of reduction in viral, they didn't really say it this way, but that's how the graph looks, as a reduction in viral set point in the folks who got the vaccine regimen compared to the placebos. And what it really was is that fewer of those folks who got the vaccine regimen ended up going back, meeting their ART restart criteria that they had specified. But they definitely all rebounded and had, kind of varying levels of viremia during that period of time. So I think it was, favorable, maybe sort of keeping the hope alive for therapeutic vaccine approach, but it wasn't a sort of slam dunk, I would say. And they did mention there's another trial that is doing the same vaccine regimen, and then it's combined with the TLR7 agonist to maybe induce more antigen expression, so the T cells that they are generating with the vaccine have more of an effect on the viral reservoir, although Brad may disagree with that.
Well, this seems to be at least the first I can recall that a therapeutic vaccine for HIV has had any sort of effect that was a positive step forward. Is that a fair assessment? I mean, it's the first time I've heard something where it's like, oh, this actually worked. Usually it's like they've tried it and it was well tolerated, but it didn't really do anything. This was at least doing something.
That's a great point, Michael.
I'll just say there was a I can't remember the authors or anything, but there was a dendritic cell based vaccine that reported a reduction in viral load. I can send around that link if I can find it as well. I think that's one previous precedent. And just to mention the HTI approach, there is also a group testing that in a dendritic cell platform that's out of University of Pittsburgh, trial called DC04, just as another example of what's out there.
Oh, thank you for that.
I mean, the issue with therapeutic vaccines still remains that the alternative is full suppression on ART. So therefore, the appetite to keep a viral replication going to a lesser degree than those that did not get a vaccine scientifically is interesting, but operationally is dead, because you can be on therapy. So as opposed to keep a viral load of 20,000 rather than 50,000 or 60,000. So from a scientific perspective, it gives us an insight into what may be components that we can build upon. But that's a far cry from saying that we have a strategy that would replace antiretroviral therapy.
That's a great point.
So, yeah, I think that is a really good point. But I think it's probably true for pretty much, maybe except for the CRISPR approach, but for almost everything that we have right now, is that, we might need to start thinking about successive strategies. So something that's not completely successful in preventing rebound or causing remission, but has a, partial effect, then, an individual might go back on antiretroviral therapy and then have another intervention approach, either the same one or a different one. And it may be sort of this successive approach that kind of eventually you get down to that level where the virus can no longer rebound.
And so we had a follow-up question specific to Brad. Brad, you mentioned the dendritic cell platforms, and people are asking, why does this matter? How is this distinctive from the platform in the HTI vaccine? Can you tell?
Thanks. I was just typing an answer, but I'll verbally give an answer as well. So dendritic cells, these are the cells in your body that have the job of initiating an immune response, initiating a response to a virus, for example. So they're very powerful at doing that. So the dendritic cell approach is kind of a custom approach. It involves a leukapheresis from the study participant. It then isolates these powerful immune initiators, these dendritic cells, from these people, and then gives them these targeted regions of the virus that we think are vulnerable, these HTI regions, and then puts those cells back into the study participant. And so that primes really a particularly strong immune response in some cases. It depends a bit on the details, but in some cases it's very powerful. So that has advantages and disadvantages versus viral vectored approaches, which is the other type of vaccine that we were discussing. So in viral vector approaches, you have the same vaccine, you can give it to everyone, and in some cases it primes a strong immune response also, but there are differences in the flavor of that immune response, and we don't fully understand what it is that is needed to make the immune response effective, but we see that bit of a signal with the dendritic cell studies. And so that's why we kind of want to follow that forward and see if we can improve upon that further.
Thank you.